Hey everyone, good evening. I'm making this recording for my biochemistry students because we've already started a project. In fact, we're about a month into it, and most of you have already even made this protein. You've expressed it. You haven't purified it yet. And we are going to go through all those future steps. I promised you I would tell you why we're doing it, and I promised you that I would make it worth your while, because this is not just an ordinary lab experiment. This is an actual experiment. This is more like an apprenticeship than it is like a by the numbers, paint by numbers kind of protocol. The protocols that we're following were actually developed from my postdoctoral work over at the Fred Hutch, and so this is how we made proteins over there, and now I'm bringing them over here to make proteins. In fact, my postdoc advisor was the one who started me off on these particular proteins. He worked on them, and then basically he said to me, hey, you want to go for these? Go for it. And so that's what these are. These are Siderocalins is what we are working on, although we don't know exactly what these proteins do, and that's part of the fun of this. You're going to be making a protein, and we don't know what it's going to do until we purify it and take a look at it. Literally take a look at it. So let's talk about what we're doing. Those of you in the class know that we're already expressing proteins. You've already grown up some E.Coli. You put DNA into them. Then you've expanded the culture to a large volume. You've added something to induce protein expression. At the end of the day, or at the end of the overnight, you spun that down, and now we have pellets waiting in the freezer. We're going to purify protein out of those pellets, and the protein that we're going to purify is a version of a Siderocalin. These are Siderocalins from different species, therefore they are orthologs, because they are located in different genomes, different organisms entirely. And so we think we know something of what they do, but this is a very versatile protein family, so there's a lot of mystery about it. So I'm going to divide this into two parts. The first part will be how lipocalins of unknown function became known, the story that brought us to the point where our project began. And it starts in my postdoc advisor's lab. Actually, before I started working there as a postdoc, it starts all the way in the last millennium. In 1999 is when some of these were done. So let's talk about that. The subject that Roland Strong was studying was lipocalins. He's at the Fred Hutchinson Cancer Research Center, and actually the immune system has a lot to do with cancer. And so he's studying immune system proteins. We worked on all sorts of things. I actually worked with Roland on a project while I was still a grad student in another lab. And then I decided, hey, this would be a great lab to do a postdoc in, and that's where this all happened. And 2 years after I was a postdoc, I got my job at SPU, and then I've been here ever since, 2003. So this is way back in the day, before most of you were probably ever born. But Roland was working on a class of proteins called lipocalins. And you can break that down. Usually you can tell what a protein does by breaking down its names. They bind lipids, which are hydrophobic things, and you know that's a very broad category. The suffix calin comes from calyx, which actually means cup or chalice. So in a sense, this is like going after, it's not quite the Holy Grail, right? But we are going after what cups hold. These are protein cups that have evolved to bind different things. Here's an example of one. The blue is the protein, the red is the thing that it binds, and you see it sits in the middle of it just like the contents of a cup. So we started with the lipocalin that we knew it was a lipocalin, we knew it was a cup, but we didn't know what it was a cup for. So they called it by the name of NGAL, neutrophil gelatinase associated lipocalin, because they knew it had something to do with neutrophils, and that's basically the vague enough name that we didn't know what it does. When we knew what it does, we actually changed its name. And so this, you can tell this is an early paper, and you can see it was submitted in 99 and published in 2000. And so Roland and his student Dave Getz were working along with others, and in fact I know most of the people on this. They published a structure of a lipocalin that was part of the human immune system. So it was the cup that they were studying, but the cup was empty. And they had to figure out, what does this empty cup bind? Here's one of the things that they showed. They showed 4 different lipocalins. The one in the upper corner, NGAL, is the one that they were studying, and they put in it what they thought might bind into it, but you see it doesn't fit very well. They weren't very happy with this, but they had to put forward some sort of hypothesis. They compared it to 3 other lipocalins. So the northwest is the lipocalin that we're looking at. They got the structure, and there's the structure right there. It's got a big blue hole in it. And that means that something big and red, big and negative, will be binding into it. So you compare that to the other lipocalins, and they mostly have smaller cups in them. If you look at that, the ERBP is a retinoic acid binding protein. That's what the R is. And retinoic acid, you might recognize retinol, which comes from beta-carotene. So I've made it orange. It's not actually orange, but it actually could be colored because it's just beta-carotene cut in half. This is actually a very important molecule to development. You'll find out a lot about it from cell biology. So the one in the northeast is a lipocalin that binds a true lipid, a fatty acid palmitate. And you can see it just sits in there. It has sort of a white pocket because it doesn't need charges to bind palmitate. And then finally in the southeast corner you have the NP protein, which is a lipocalin that binds a heme histamine complex. And that's an interesting thing, but you can sort of see the flat heme ring sitting right into that flat groove that is made to bind for. So looking at all these things, the only thing you can be really sure about is whatever NGAL binds, it's not one of those 3 other things. It's something new and different, something big, and something negative. So they made their measurements of the open calyx. They looked at the size of the groove. They compared it to the other ones. And just remember, there's a side view of the pockets that's binding something. The retinoic acid is just a little skinny molecule, and sort of it sits into the binding pocket, complementary interactions. That MUP protein, it actually stands for mouse urinary protein, but that's binding something that's even smaller. It's 2-sec-butyl-thiazoline, and in fact, I don't even know what that is. I'd have to look it up. Whatever they are, we know that whatever NGAL binds is probably bigger, and the thing that they were putting into it, the NCA group that you see up there, doesn't quite fit, and they were very clear about that. They were very clear that's just their best guess, but they really didn't know. They needed some lucky break in the lab to be able to figure out what binds, and it turns out that Dave had one of those classic lucky breaks in the lab. One day Dave was making the protein. Usually we make them in BL21 cells. You all are familiar with that one. But he was actually making it in DH5 alpha cells. I honestly don't know if this was a mistake or deliberate. DH5 alpha cells are usually used for cloning. They don't have all this machinery to make as much protein as they can. They're just normal cells. They make a lot of DNA, not a lot of protein. But he put them into that, and when he purified out the protein, he saw what's this right here. He saw it was bright red. So he immediately started saying, this is the clue we've been waiting for. It binds something that is this bright red, rusty color. I wonder if it could be iron and oxygen of some form. In fact, iron is very important to bacterial systems. Therefore, it's important to the immune system because the immune system wants to mess up bacterial systems, right? So that's their big clue. This comes from their next paper, which I believe was a 2002 paper. The next paper that comes out tells you what the protein binds. And only later, after that paper, did they officially change its name to what it binds. It binds siderophores, and sidero means iron in Latin, I believe, or whatever language it comes from. And so this is an iron-binding protein, but it's not just iron binding, it's binding iron plus another molecule. Siderophores are what it binds, and siderophores are little molecules that bacteria make. They're like little claws that grab onto iron. So the bacteria make this little claw, and they're trying to grab the iron for themselves because they need it to survive and to invade your body. Of course, the immune system doesn't like that, so it's adapted a lipocalin to be able to bind the whole claw, the siderophore iron complex. And it turns out that those tend to be negatively charged. So orange, negatively charged. So it's this is clearly a part of the immune system that is working against bacteria by taking away their iron from them that they're trying to get. It's a little complicated because it's a 3-layer molecule. Iron, siderophore, siderocalin. But that's held up with the standard of time. So because it's red, we know it absorbs in the visible wavelengths. It absorbs something around, it absorbs blue or yellow light because it lets the red light through. And in fact, that's exactly what you see if you put it into a spectrometer. You see that it's low around 700, that's where red light is, but around green 500 and blue 400 nanometer light, you see that it's absorbing. The only thing that doesn't absorb here is when you have the empty or apo N-gal protein with nothing in it. It's a clear, regular protein just made from amino acids. There's nothing in there to absorb visible light. But when you put the iron in, then it binds to the protein and it sticks with the protein, turns the protein orange. Big clue that the protein is doing this function, and we figured out how it works. I'm going to want you to do stuff like that because we have visible spectrometers. Once you make this protein, you can actually look at it that way. And so I'm wanting to know what you see, and that's what we'll be doing down the road. Another thing we can do is that Dr. Pierce manages a atomic absorption instrument that we have upstairs. And that actually can measure quantities of transition metals. So they used one of those, and they measured how many moles of iron was in a purified red sample. And it turns out there was exactly 1 mole of iron for every mole of protein. 1-to-1 molar ratio, it all makes sense. What I'm thinking with this is I'd like to replicate this, but this is another experiment down the road. This is something I'd like to replicate in the spring in my Survey of Physical Chemistry class. So if you take that class, by the way, you'll get more research experience, and you'll be able to put that on your resume and tell people that you've done it. Okay, so now we've actually defined what that is, and actually I had a picture of it from the very beginning. We now have pictures of crystal structures of enterobactin bound into the Siderocalin calyx. And that's exactly what this is. There's a picture of the enterobactin. You see all those oxygens that are coordinating. The red oxygens are coordinating to the orange iron in the middle. That's how the bacteria grabs onto the iron, so we grab onto the grabber. Now, there are a lot of different bacteria. Bacteria make a lot of different molecules. It's whatever works for them, right? And so you can see at the bottom of this screen, you can see enterocalin or enterobactin, which is the molecule that we showed you binds siderocalin on the last slide. But notice that even small molecules, these other molecules on it also bind. Notice the molecules above it don't even have to have all 3 rings joined together. They have to have 3 rings, by the way, because each ring sticks to the iron twice. I mean, look at that right there. Each ring is coordinating to 2 parts of the iron. The iron likes to coordinate to 6 oxygens, so the bacteria makes a siderophore that is very happy to oblige. But those 6 oxygens don't have to be all on one molecule. It looks like this broken molecule will actually bind iron, and then will bind the siderocalin. And even that molecule on the top, it's a different bacteria making a different siderophore, but it binds iron just the same. And it turns out that our siderocalin is able to bind this as well. So it's an interesting question. What are the limits to what the siderocalin recognizes? How weird can the siderophore get? You have all these other things that it binds, and you can see there's a trend here. You have 3 rings, so it's got to have 3 pockets to bind the 3 rings. And it's got to have positive charges to balance all these negative charges. But other than that, it does have flexibility, which means that it's good for us because it means that the bacterium can't just make a slightly different structure of siderophore and then grab all the iron at once. It can't get away from us that easily. Our siderocalin is flexible and able to bind all these different things. So when we look at the structure of siderocalin bound to enterobactin, which is shown right here, you can see that it does have exactly like we expect. It has 3 pockets down there. They're numbered 1, 2, 3 up there. And you can see each of the rings, each of the 3 rings sitting into that pocket. And it all fits together. It's like a big jigsaw puzzle. By the way, each of those rings is called the catecholate ring. That is the oxygen going off of the ring structure that it has. So we'll use that term, but we can go down and we can even say which amino acids in the siderocalin are binding to the different parts of the siderophore, the enterobactin, or the carboxymycobactin, which is the other one, and we can say how do they do this? And it actually uses some of the same and some different amino acids to be able to interact with the 2 different siderophores. So it's kind of cool how it does this, and it's an interesting question. How does it manage this balancing act of being able to bind all these different molecules to be able to defend your body against different bacteria? And there are other stories because each different species has a slightly different siderocalin, and some of them can bind some bacterial products and some of them can't. So that means certain bacteria will be able to infect certain species because of their siderocalins that they have. You see how this is all coming together to tell a very interesting story. So here are a bunch of different siderocalins, human, rat, and mouse. And then there are other lipocalins that you go across. The numbers are kind of small, but mostly I just want to show you, if you can squint or zoom in and look at the numbers, you see that if something is 60% identical, it's still a siderocalin. It does the same thing. If it's 30% identical, we start to wonder. Maybe it binds siderophores, maybe it does something else. Or maybe it even does two things at once. And we have a story like that. So we actually have a structure of the chicken X-FABP. In fact, FABP stands for fatty acid binding protein, which makes sense. A lipocalin binds fatty acids. So a good name for it is FABP because fatty acid binding protein. But where do the fatty acids bind? So the chicken X-FABP is one of the proteins that we actually have a structure of. Of course, as many structures as we have, we wish we had more. We don't have enough to really know all the genes and what they're doing. But that's why we have new proteins to make. The protein that you're making is either an important protein that we need to study, or it's a protein no one's ever made before. So we have a lot of good work to do. So here's a story about chicken X-FABP. It has a slightly different groove. It will still be able to bind most siderophores, just like, and you see it has 3 pockets and negative charge down there. But look at that thing off to the side. That's sticking out, and it's a sugar group. It's able to bind a glycosylated siderophore, because it turns out a certain species of bacteria that infects chickens has figured out that its siderophores are being snatched away. So what it does is it takes a sugar group and it glycosylates its siderophore to make it a different shape. It doesn't care about the different shape, but the other siderocalins do, because it won't fit into them. Well, then the chicken has a response to that, because the chicken mutates a groove that will be able to bind the newly glycosylated siderophore. So the bacterium was able to infect the chicken, then the chicken builds a new siderocalin, and then the bacterium can't infect the chicken anymore. And we actually do show that the infectious nature of the bacteria that have this actually follows this exact plan. So you can see, you can trace the arms race between infection and infected by watching what's going on with this siderophore and the siderocalins that bind it. Another cool thing about chicken X-FABP is that actually, if you look down, it's got a big cavity underneath the regular binding cavity. You see right there in part C, there's the enterobactin bound at the top. Underneath it, there's a phospholipid. And so we don't know exactly what this is doing, but it's clearly supposed to be there because, number one, the cavity is the right shape, and number two, it has all these positively charged residues that hydrogen bond and form ion-ion interactions with the negative phosphate. So it looks like it has a second binding site, and we'd like to figure out what it does and if it's significant. Maybe it's just there, but it doesn't look like it's just there. And so that's one of the surprises that we can find out with these proteins. This is another one called alpha-1 microglobulin. I swear I changed that, and the computer keeps taking it back. It's microglobulin, not macroglobulin, I believe. But this alpha-1 microglobulin binds heme. It's a siderophore that binds heme. So if you take a slice through the crystal structure, and you just look at it from the side, you see, number one, negative pocket, because it's, or positive pocket, because it's blue. It binds a negative thing. And it's sort of a narrow, deep cleft that will bind the heme. You can imagine the heme just sort of sliding in there like a card into a reader. And what's even cooler is that some lipocalins, they don't just bind lipids, they actually catalyze them and they react them. Prostaglandin D synthases will actually take a lipid and they will change its bonds around to make it into a signaling molecule called a prostaglandin. It's a class of lipids, probably came up in your lipids chapter in 4361. And in fact, so here's two different enzymes that do the same activity. You see they have completely different folds, but the one on the left has a lipocalin fold. So it looks like the lipocalin evolved to bind a lipid, and then it evolved the activity, and it just takes a couple of mutations to be able to have a catalytic residue that's in the right place, to be able to catalyze the transformation of the lipid into a prostaglandin signaling molecule. Very cool. So it's possible that one of the things that we're purifying might not just be a binding protein, it could actually be an enzyme. Wouldn't that be cool? So that's why I want you to look at these things. I want you to find out if that is. This is the actual, if you want to know more about what the enzyme is like, this is what the enzyme does. You see there's a lipid at the top, arachidonic acid, and it gets turned into all these prostaglandins at the bottom. The PGH2 into PGD2, if you look down at the very corner, the very lower southwest corner of the screen, that's PGD2. And you see it just has a bond that's been made, and maybe it has some oxygens that have been added. So it's pretty easy to turn arachidonic acid into this prostaglandin, and it just takes a couple of residues, and it looks like those couple of residues were added onto the lipocalin, and it made a new enzyme. And it's involved in regulation of pain, REM sleep, and this has been shown in mice at least. It looks like it's a pretty important molecule. And that's our little lipocalin. We can't rule out that one of the proteins that you are purifying may have a surprise enzymatic activity like that. Actually, the question is, can we detect it? This is what the reaction is that it actually catalyzes. So that's a lipocalin that's able to do that. And in fact, even more than that, the surface loops of the lipocalin, they open and close depending on what the enzyme needs to happen. And so that's where, if you look up there, you can see some movement depending on whether the loop is over one way in the pink and over another way in the blue. And that's the kind of structural study that we can do down the road. That's why we're making so much protein, because who knows what kind of study we'll want to do with it someday. If you look down at this is the enzyme structure, and you can find the catalytic residue, and there really is one key residue. There's just a C65, a cysteine, in the right place. Once it binds a lipid, it's able to catalyze the reaction. Very cool biochemistry. So that's part one. That's what siderocalins can do. And so we're starting with a human lipocalin that binds multiple siderophores, and we're making different lipocalins that are related to that. We think that they'll probably have some iron binding, siderophore binding activity, but we're not really sure about anything. But we know that something's up because we literally seen that there is something different about some of these proteins that we've made, and we're trying to replicate some of that this year. More about that when we get to part 2.